Okay, welcome back to another Poulter's Pod Year 10 video. We're going to continue with our work on preparing for our paper 1 next week, and we're going to look at the B3 infection response topic now. So in this topic, only a few bits are going to come up in that test next week. One is going to be about bacterial infections. We also need to know about plant deficiency diseases and about monoclonal antibodies. So let's start by looking at bacterial infections. So, first of all, you should remember how bacteria make us ill. Bacteria make us ill and give us symptoms because they A, produce toxins, and B, they reproduce rapidly. As their population grows, the more toxins they make, the more symptoms we're going to get from them. You have to also be aware of two specific bacterial infections. One is salmonella, the other is gonorrhea. So look back and think about what the classic symptoms for those two bacterial infections are, okay, and how they might be spread from person to person and how we can control them. So have a look at your revision guides and your notes, etc. for that. All right. Another thing that might come up here is about, testing antibiotics and how they might affect bacterial growth, and we can use them to test the effectiveness of antibiotics or disinfectant. So the classic one is this: you would set up a petri dish with a bacterial colony using your various aseptic techniques. Make sure you sterilize the surface, you sterilize any equipment, you keep the lid on the petri dish as long as possible to prevent any airborne pathogens contaminating it. You might want to sterilize your inoculating loop under a Bunsen flame to kill any contaminant microorganisms there as well. So I've added my bacteria to this plate using aseptic techniques to reduce any potential contamination. And then what I might do is add little discs of my antibiotic or disinfectant depending on what I'm testing, like that. I would then leave this to incubate at 25 degrees C in a school environment. The reason I would do it at 25 degrees C in a school environment is to prevent any kind of pathogenic bacteria. Remember, these pathogens are microorganisms that cause disease from growing. If I grew it at 37, that might provide ideal conditions for pathogenic bacteria. It'd be far too dangerous. I've now given it a week to say incubate, and then I could compare what I would call the zone of inhibition. This is the area around each disc where there is no bacterial colonies, effectively where the disinfectant or the antibiotic has killed the bacteria. So you would see these sort of circular regions of various sizes around your discs. And looking at this simple example I've just made up here, this one has got the largest zone inhibition, it's got the largest diameter, and I might have to, pi r squared and all that kind of stuff to work out the area. I can say that that one is the most effective, whereas that one there is the least effective because it's got the smallest, in this case, no zone of inhibition. All right, so that'll do there for bacterial infections. You guys are going to need to make sure you go and look at the symptoms and how they spread as well on top of that, so when you're doing your revision work. Let's move on to now plant deficiency diseases. Okay, so plants, like human beings, can suffer from deficiency disorders. In human beings, if I'm lacking certain key nutrients or vitamins and minerals in my diet, I will develop deficiency disorders. A classic example would be a lack of vitamin C in my diet would lead to scurvy. A lack of iron would lead to anemia. Plants have a similar problem as well. Plants need certain key mineral ions from the soil, which they typically get through active transport. The two that you need to be really aware of is, number one, magnesium ions, and the other is nitrate ions. Magnesium ions are used to make chlorophyll, which allows the plant to absorb light energy for photosynthesis, so it can make glucose for respiration and growth. The other is nitrate ions, and what that plant uses them is it combines the nitrate ions with glucose to make protein, make amino acids, then build our protein. Without the protein, the plant can't grow sufficiently. So what would I look for here? So one of the key things is signs and symptoms of a nutrient deficiency. So if a plant is not getting enough magnesium, it can't make chlorophyll. and we know that chlorophyll was green. So one of the classic symptoms here would be yellowing of the leaves, and as a result, less chlorophyll, less photosynthesis, possibly stunted growth as well. These are sort of signs I would look for. Nitrate, because it's linked with protein formation and therefore growth of the plant, I'm definitely going to see stunted growth as a main symptom there. So factor all those sorts of things in there, make links to the processes that are involved with these ions and the symptoms and signs you would get for these plant-based diseases. Okay, so let's move on to now probably the more tricky part, the monoclonal antibodies. So monoclonal antibodies have been engineered by scientists to help us diagnose diseases in animals, plants and humans. We can use them in pregnancy testing, and we can use them to deliver drugs to cells such as cancer cells. The one thing I want to focus on is actually how we go about mass producing monoclonal antibodies, because you might need to know the actual methodology behind it. So first of all, you would take an animal like a mouse and you would inject it with a protein that you want the monoclonal antibodies to actually bind to. Now that protein could be an antigen from, say, a virus, or it could be a certain hormone like we want to look at in, say, pregnancy testing, whatever it may be, even from a plant even, okay? Doesn't matter. Just get that protein and inject it into a mouse. That mouse is lymphocytes, it's specific types of white blood cells, try and use that term lymphocyte, will then start to produce antibodies to counter that particular protein. We would then take those lymphocytes from the mouse, we'd take a blood sample and collect them, and we would then fuse them with tumour cells. We call this a hybridoma, so it's basically a combined lymphocyte and tumour cell. We would then clone that hybridoma, which will then divide very rapidly and produce antibodies. Now they're the monoclonal antibodies we want, so we can mass produce them for our particular purposes. So an example might be, I want to test for coronavirus, so I would isolate one of the proteins, one of the antigens on the coronavirus. I would inject that into a mouse. The mouse's lymphocytes would then produce antibodies for that protein. I would then harvest the lymphocytes and combine them with a tumour cell to form a hybridoma. I would clone that hybridoma, which would then divide rapidly and produce the monoclonal antibodies for coronavirus, which they could use in a coronavirus test. Pretty straightforward. Just related to the context of the question you're doing. The same things always apply. So that's it from me. This is Dr. P signing off. Have a good one. Cheers.